Have you ever wondered how scientists can separate DNA, RNA, or even proteins with just a jiggly jelly-like slab and some electricity? This is gel electrophoresis, a technique so powerful it's used in everything from crime scenes to cancer research. And today, I'm going to break it down for you in the simplest, clearest, and coolest way possible. What is gel electrophoresis? Gel electrophoresis is a lab technique that separates charged molecules, like DNA, RNA, or proteins, based on their size and charge. Think of it like a molecular racetrack. Smaller fragments run faster through a gel, while bigger ones get stuck and move slowly. Electricity is applied across the gel, pulling the negatively charged molecules toward the positive end. The result? Band to tell a story of identity, size, purity, or even mutations. Types of gels: Agarose versus SDS-PAGE. So, not all gels are the same. If you're working with DNA or RNA, you'll likely use Agarose Gel Electrophoresis. It's like the bread and butter of molecular biology. Simple, cost-effective, and perfect for nucleic acids. But if you're studying proteins, then it's SDS-PAGE, a polyacrylamide gel system. It denatures proteins and gives them a uniform negative charge using SDS detergent, so you can separate them based only on size, not shape or charge. Real-world applications. You're not just running gels for fun. This technique has real impact. Forensics, DNA fingerprinting to solve crimes. Medical research, protein separation to detect diseases. Genetic engineering, checking plasmid digestion. Biotech industry, purity testing of recombinant proteins. How it's done. Here's how a gel run actually works. 1. Prepare the gel. 2. Load your samples with dye into wells. 3. Connect to a power supply. 4. Watch your samples move through the gel. Stain and visualize the bands. Boom! You just separated biological molecules with science and electricity. So whether you're cracking DNA codes, checking your PCR results, or purifying proteins, gel electrophoresis is your best friend in the lab. If you found this helpful, smash that like button, share it with your science gang, and don't forget to subscribe for more biotech breakdowns every week. Leave a comment below. Have you ever run a gel? What confused you the most? Subscribe and follow at Funtrest Bond.